Okay, right, okay.
Okay, it's good.
Yeah.
So you can.
Yeah, I can go now. Yeah.
No, it's okay.
Great. Yeah.
Okay. Hi. Thank you. So it's my great pleasure to introduce Phil Mullineaux.
Yeah.
He's in our lab for 4 weeks as a visiting scientist, kindly supported by Judy. And it has been very productive. And so he's coming from Essex University, but he originally is a virologist. He has been starting his career in virology.
My postdoc career.
Postdocs, and then he got in a long time in Norwich in the John Innes Center. So then he moved many years ago in Colchester, where he got appointed to the professor position, and he's one of the most productive scientists in the field of redox signaling and antibody signaling. So, Phil, the stage is
well, thank you very much. I thank you to Tulip, the Tulip Consortium, for funding this short and, as Jean-Philippe said, has been a very productive visit. I'll only very, very briefly touch on that right at the end of the talk, sort of the rationale for the visit. So I'll leave you with the title there, so I'll move straight on because time does not wait. I've been interested in the role of reactive oxygen species, in particular one, hydrogen peroxide. Yeah, for those of you, maybe chemistry is not so good, but also I'll be talking about another one, which is not a reactive oxygen, not an intermediate, a reduction intermediate between water and oxygen, but an activated form of oxygen called singlet oxygen, which is actually a very potent causer of oxidative stress in plants. I'm particularly interested in the role in which these play as signal molecules going from the chloroplast to the nucleus. And this has been a story of stop, start, stop, start, partly because of the way UK funding works, but also partly because we had technical limitations, which really inhibited the testing of our hypotheses and ideas. And it's only actually towards the end now of my career, I've just retired, actually. So just at the end of my career now, where actually we've now got some of the technology we can use to really probe the story. And what is revealed so far, even in only several years of work, has been really quite profound, I think, in terms of the way we think about hydrogen peroxide signaling as a signaling molecule, as a signal transducer between chloroplasts and nucleus. So it's signaling. Oh, I should just point out that there's lots of signaling molecules derived from candidate signal molecules derived from singlet oxygen. I'm really just going to concentrate on the very common one, which occurs right across all aerobic organisms. That singlet oxygen gives rise to a considerable amount of lipid peroxide production, especially from membranes, especially from fatty acids and phospholipid bilayers. One of the reasons I've been interested in this, and it's to plug into a larger group of the photosynthetic physiologists who are very much interested in the processes that affect photosynthesis, both at the very short term responses to, say, changes in light intensity, which is the most convenient way of doing this, really very rapid changes, all the way through to gene expression changes, which was my interest, and the fact that reactive oxygen species tend to sort of be involved in about that time scale. And for a long time interested in transcriptional changes, but now I must admit, I wish I'd spend a little more time on studying the actual proteins, as will for my stay here in the last few weeks, all the way through to acclimation, adaptation, and even adaptation speciations, all a continuum. So it's how these reactive oxygen species, how far does a change here manifest itself very way down this time scale. Now, this all starts with some relatively simple stories, which is really on the right side of this slide. This is just me tinkering around with one of our latest ideas about the integration into cryptochrome signaling. But the there's some very old, long-standing observations that if you pre-treat a leaf with hydrogen peroxide and then subject it to a substantial increase in light intensity, you prevent you can slow down or ameliorate photoinhibition. That is the inhibition of photosynthesis, which is triggered by actually reactive oxygen species and damage to the chloroplast photosynthetic machinery. There we are. Conversely, and paradoxically at the time, you treat a leaf with glutathione or scorbate, two water-soluble powerful antioxidants, very common. You create increased susceptibility to photoinhibition. And a lot of people had these data and never knew what to make of it. We're going back to the early 1990s, when the notion was, basically based on biomedical studies, that free radicals and reactive oxygen was bad and antioxidants were good. And therefore, the simple premise was, me coming in as a genetic engineer, that if I increase the antioxidant content of a plant, we're going to have wonderfully resistant plants, save the world, and everything will be fine. No. We spent a lot of time increasing the glutathione content of the chloroplasts and the cytosol of tobacco. And every time we did it, this was the outcome. We actually got increased oxidative stress, programmed cell death, in proportion to the degree of light intensity that was used. And if we tried to grow these plants, for example, in a greenhouse, they were sterile. They were completely sterile. So, in other words, what we've done, as it now subsequently turns out, we'd desensitized the plant to the changing environment, and the fact that the chloroplasts are used as a sensor for what was going on in the light environment and what was going on in relation to other components, such as temperature, such as what was going on inside the cell. So this started to embark us on a long journey, which, as I say, is a stop-start journey. But I'll take you through some of the key points. One of the first results we got was with using the idea that we found a marker gene which was really highly responsive to changes in light intensity associated with photoinhibition. It's called the APX2 gene, and it's been very well used in the community over many, many years for this and for heat stress too. And we were able to fuse it to luciferase, transform Arabidopsis, which at the time was not a trivial exercise. And we were able to show that, in fact, hydrogen peroxide was a systemic signal under abiotic stress conditions. It had been an influence for plant pathogen interactions for some time, but this was the first showing of how it might work for abiotic stresses. And then other laboratories have picked up, mainly Barry Pogson's lab and Ron Mittler's laboratory in the States, were able to show that it wasn't just hydrogen peroxide, but calcium and electrical signals involved in long-term, long-range signaling. It involved a sort of cascade effect, or a pass the baton from one cell to the next during this process of signaling spread. But I deviated from my colleagues there, because I actually went off in a different direction. I wanted, after that work, to actually ask the question, well, what happens inside a single cell? How does that hydrogen peroxide work? And actually, when you start to think about it, there are some actually pretty fundamental problems. First is, it's not only chloroplasts that make hydrogen peroxide, but hydrogen peroxide comes from a number of other sources. The proteasome, especially in C3 plants. You've got mitochondria can produce hydrogen peroxide, not listed here, but there's the plasma membrane, and reactions in the cytosol, and so on. And the question is, the nucleus, if it's going to respond to hydrogen peroxide responsive genes, of which there is plenty of evidence that they exist, how does it discriminate? How does it manage to know that hydrogen peroxide, say, coming from mitochondria is different from hydrogen peroxide coming from chloroplast? The second problem is as you get to know your biochemistry more, is that the environment, the cytosol environment, if hydrogen peroxide was going to be a signal transducer, how on earth would it cross a reducing environment like the cytosol? And third, and perhaps the most subtle, but probably the most difficult question is, of course, we've all seen review diagrams like this. A single chloroplast communicating to the nucleus, for example. But in fact, that's not the reality. The reality is there are many chloroplasts inside the cell. And in fact, in a varied environment, they're all in different physiological states. So how on earth do different chloroplasts emitting different signals, how does the nucleus make sense of that in relation to integrating those signals? So much so that for a long time, I proposed and others proposed theories which effectively said, oh, hydrogen peroxide, say in the chloroplast, must be converted to compound X, which is specific for that, which then migrates to the nucleus and does various things. Turns out that's wrong. But for many years, we tried on that basis to get it to work. The breakthrough came with the seminal work in which from Belousov and colleagues, the Russian scientists, who developed the first of a series of biosensors for hydrogen peroxide based on the circular permuted YFP. And in this case, the hydrogen peroxide binding domain of a transcription factor from E. coli called OxyR. You put these together in a certain way, and when OxyR binds hydrogen peroxide in a thiol-dependent reaction, it changes its conformation. That conformation change is transmitted to the circular permuted YFP, which then undergoes a change in its fluorescence characteristics. Now, the key fluorescence characteristic, and I'll mention this more than once, is that there are two, and this is essential for plant work, there are two, one emission, one wavelength emission at 516 to 530, a very wide band. There's actually two excitation maximum, round about the 400, 405, 480, 500. And it means that what you do is you express, if you express the emission values from each of those excitations as a ratio, then the changes in that ratio are directly proportional to the oxidation state of the probe. And if the oxidation state of the probe reflects the degree of steady state level of hydrogen peroxide. And it's independent of probe concentration, and that's the key factor. Otherwise, you have to account for that too by other forms of mathematical and experimental approaches. So, and you can see here the sort of this one, and there's been various other ones that have different dynamic ranges, but this is the sort of range you're dealing with. I should add that it is a steady state that's been described because the re-reduction of the probe occurs with the cellular glutarreduction and glutathione systems within most cells. So there is some variability there, but nevertheless, that's the way it works. Very briefly, I'll just mention that we use a lot, although we use Arabidopsis as well, but I'm going to mention primarily work using the Gaussiana-Benthamiana transit expression systems because we can turn around lots of experiments very quickly, and it's very compatible with working with confocal laser microscopes, in which you can actually do the highlight stress of various treatments to your leaf discs actually on the microscope staging. And we find this a particularly useful system. So I'll say no more to it than that, but I can obviously give details for anyone who wants more information. It's nice highlight stress because you and the oxidative stress of that promotes, because you can monitor the actual, quantify that, and we use chlorophyll fluorescence. And we use the simple parameter FV over FM as an imaging system. And if you stress in our type of system, where you're giving about 1,000 micromoles of light intensity against the plant grown at about 200 to 100 up to 200, depending on what you want to do, then you will get a signal drop like this, which is a good indication of photoinhibition. But if you then leave the plant 24 hours and measure it again, it's recovered. So it's what's called as reversible photoinhibition. And that's good because reversible photoinhibition is particularly associated with the accumulation of hydrogen peroxide and not singlet oxygen, which I'll mention in the next little section. And just to show, if you do a grind and find Amplex red method for detecting hydrogen peroxide, you see an increase in the amount of total hydrogen peroxide. Okay, now the paper that really helped us a lot that we did, that we published in 2017, has a lot of data in it, but it actually exploded the idea that the hydrogen peroxide somehow was converted to substance X, which then went to the core. So there's two slides here. I'm just going to extract from the data. I just want to talk you through it. oxidation state of the hyper is indicated by the black line here. Now we put the lights on the leaf, up it goes. Now the one problem with hyper, or these earlier versions of hyper, is that it does respond to pH. So you have to do a control, you have to build a control, which we call cipher. And cipher, excuse me, cipher is this orange light here, and you can see that it's not undergoing a change. There's a pulse at the beginning that was expected because of alkalinization of the stroma as you hit the plant with more light. Okay, so far so good. We then co-expressed, this is why the transient expression system is nice, we co-expressed a stromal ascorbate peroxidase. This is an enzyme which will catalyze the reduction of hydrogen peroxide using ascorbate as the electron donor. And you can see that we damped the signal. So in other words, yes, we are measuring hydrogen peroxide here. It's related to hydrogen peroxide, the oxidation state of the probe. So that's good. All quite conventional, no big surprises here. And then this is using a much loved herbicide in our area called DCMU, which inhibits, when applied at this stage for this length of time, about 1 hour, is an inhibitor of photosynthetic electron transport. And you can see again that it completely damps the signal. And so we could conclude, yes, hydrogen peroxide is made in the chloroplast stroma in response to increased light, and it's dependent upon photosynthetic electron transport. No big surprises there. The big surprise comes in the next slide. The one. Right, okay. This time, and this is going to be about the flexibility of our experimental system, we moved the hyper to the nucleus, and we targeted it there. But we left, just remember, we left the stromal APX in the chloroplast. Okay, we didn't move out of the center. Now, again, you switch on the highlight, boom, hydrogen peroxide levels go up in the nucleus. Aha, good, interesting. We hit with DCMU, and this is the first nice result, the inhibitor, it blocks it. So photosynthetic electron transport is required for the accumulation of hydrogen peroxide in the nucleus in response to high ion. The cipher shows us that there's no pH effects, but the biggest surprise was when we hit the stromal APX. So the APX is still in the chloroplast, and yet it blocks the signal to the nucleus, in the nucleus of hydrogen peroxide accumulation. We had to conclude that the simplest explanation might be that the hydrogen peroxide comes from the chloroplast to the nucleus. But what really threw me for a long time, through Nick Smirnoff, who was a collaborator of this work, we did co-expression with a cytosolic ascorbate peroxidase, and we didn't block the signal. So how is it we block the signal in the chloroplast, from the chloroplast, apparently? But if it had to cross the cytosol, what happened? So we had to conclude, reluctantly, that the hydrogen peroxide had somehow, if it had moved, had avoided the cytosol. Now, graphs tell you so much, but actually images are actually really useful as well. And the first thinking, the penny, as we say, started to drop about what might be going on. If you just look at the bottom slides here, here's the nucleus. And here's the hyper responding to 60 minutes of highlight. You can see the accumulation of hydrogen peroxide substantially in the nucleus. You also see hydrogen peroxide in the cytosol, but it's only little pockets, which turn out to be what we call microdomains of hydrogen peroxide. Hydrogen peroxide is not evenly distributed in the cytosol at all. All right, so there's something going on there. But when you superimpose chloroplasts onto this image by the chlorophyll fluorescence, these little microdomains here are associated with chloroplasts that are not attached to the nucleus. Those that are attached to the nucleus, of which in our epidermal abaxial cells that we look at here, there's a range of them. They don't issue any hydrogen peroxide apparently into the cytosol, but they seem, but what we would argue, or we thought, that maybe they were secreting it into the nucleus. just as a side before we go on to the spatial component, is just to say that the same type of controls we did with stromal, cytosolic APX, and DCMU, we measured gene expression for a light responsive gene. That's all we could do at the time. We used another APX for that. And basically, we could show that there was a signaling consequence to the movement of that hydrogen peroxide into the nucleus. So the idea was, oh, maybe the hydrogen peroxide is secreted into there and enters and stimulates signaling directly in the nucleus. Now, chloroplasts are free in the nucleus, but they're also associated in this particular cell type. They're easier to spot. In a mesophyll cell, which is packed with chloroplasts, it's much harder to spot that there's a specific association, what there seems to be here. So here's a sort of distribution of numbers. And if you look at the redox state of the probe in chloroplasts associated with the nucleus compared with chloroplasts that are not associated with the nucleus, the redox state of it is lower, implying that there's a lower concentration of hydrogen peroxide in the stroma of those chloroplasts associated with the nucleus. So what we're seeing evidence-wise for the first time is there is some form of subpopulation of chloroplasts. So there's division of labor of what chloroplasts could get up to inside a cell. Okay. So that takes us as far as one story does at the moment. Now, to extend the story a little about another spatial component, which is also key, we have to switch to another set of probes. These are ones that we've built along with others, and these are called redox relay peroxide sensors. And this is to overcome a couple of problems with hyper. If one wants to do various things, such as, for example, be flexible over the substrate, but also actually produce transgenic plants, because we had a lot of problem with hyper silencing in Arabidopsis when we put it in Arabidopsis. There has been some overcoming of that of late, but we were having a lot of trouble. So we switched and tried this one, and this is a tethered system where you use a redox-sensitive GFP with a redox sulfur center here, and a glutathione peroxidase, isoform of some kind, linked to it. And what happens is in this chain reaction here, the oxidizing equivalent in the peroxide is transferred to the redox, to the Rho GFP2, which then changes its characteristics in exactly the same way as we said with hyper. Okay, so you still get that same fluorescence changes. It's a better probe, it's a lot stronger. So that's one of the other things. It's also pH insensitive, which makes life particularly useful. And you'll notice that I put ROOH, not H2O2. Okay, and this is the other issue. Is it only hydrogen peroxide that is involved? Are other peroxides involved? So we designed a probe from design principles which copied or mimicked a relationship. Now, most GPXs that you find in most cells, are of the kind which will have a very wide substrate specificity. So they will degrade quite a number, or they will reduce a number of different peroxides. Hydrogen peroxide, but also lipid hydroperoxides as well. However, there is one pair that don't do this, and that's Homo sapiens, human GPX1 and GPX4. GPX4 has a wide substrate specificity, but GPX1 has a very narrow specificity, only associated with hydrogen peroxide. And if you line up the sequences around the active site domains, this is what you see. You see this insertion, what is an alpha helical domain in this section. Now, we couldn't use GPX, human GPX, in plant cells because there are selenocysteine enzymes, and there is no selenium cysteine system in plants. So we took another well-known one, the oxidative response peroxidase 1, which is also known as GPX3 from yeast, which has been implicated in signaling. And also, this construct has been around for some time. An ORP1, that is a GPX3 fusion to Rho GFP2 that reports peroxide, but incorrectly, in many papers, they say hydrogen peroxide. Okay. Now, it's understandable why. So we made a synthetic version with these insertions in. And I'm not going to show you the slide, but it just shows you this particular arrangement, you get exactly the same response to hydrogen peroxide. All right. There's nothing's magic there. The good bit comes when you take unilamellar liposomes, treat them with lipoxygenase to form fatty acid peroxides as part of phospholipids. And if you treat raw GFP2 or RP1, despite what everyone would say, it goes under massive oxidation in response to this treatment, to the addition of lipoxygenase to a liposome preparation. But raw GFP001 does not do it too much. So in other words, we can discriminate between the two. So now, if we use these in a pairwise combination, we can infer accumulation of lipid hydroperoxides as well as hydrogen peroxide in the salt. Okay. This is the key slide. The paper has just been ready to be submitted by Christmas. And I can walk you through this, but basically, first of all, each dot in the chloroplast is a chloroplast, right? So you see the sort of heterogeneity you see in the response to the system. So if you look at the exposure to chloroplast to highlight or highlight plus DCMU, now if you use DCMU with highlight for this length of time, which is 3 hours, you switch into another oxidative stress situation, in which you make a lot of singlet oxygen. And that singlet oxygen goes on, we assume, to make lipid hydroperoxides. And indeed, if you look at chloroplast is supposed to be highlight, you can see that there is an increased oxidation of the probes. But if you have the DCMU, well, there's a bit of a discrimination beginning to appear. You're seeing a lot more oxidation of this probe, which is associated, but this probe is less. So if that's the case, there's no equality here, then that means there's probably hydrogen peroxide being made, yes, but there's also lipid hydroperoxide. Well, what becomes interesting is if you look in the cytosol from the same treatment, here, there's virtually no hydrogen peroxide accumulating in the cytosol, which reinforces what we were saying about we couldn't detect anything in there. But the lipid hydroperoxide accumulates substantially. Conversely, in the nucleus, there's very little lipid hydroperoxide, but there's a strong accumulation in the nucleus. So in other words, we're getting a different separation of peroxides on the spatial and in this intercellular relationship. Now we know hydrogen peroxide is involved in signaling. We also know that lipid hydroperoxide is involved in signaling. Various ways. One is through the production of jasminates. The other one, of course, is directly through acrolein and other sources reacting electrophile species, but they all trigger the same type of pathway, which is a programmed cell death pathway, a PCD pathway. And that's consistent with if you apply 3 hours of eye light with DCMU, you will trigger a lot of tissue damage. Okay. So that's the initial work. That's where we are. What I'm now going to do is take you into where we are with some work that we've just got funded with BBSRC, which explores this spatial relationship more carefully, and at the same time also delivers us to why what is a nicely developing collaboration with Jean-Philippe is becoming crucial. So we've already raised, is there to observe subspecialization of chloroplasts? And as you say, Sally McKenzie in Penn State University calls them sensory plastids. She's already done a lot of work in this area. Of course, the other thing to point out is there's not a gap here between these organelles. There's 2 sets of double membranes. The particularly interesting one is the perinuclear space between the inner and outer nuclear membrane, which is contiguous with the ER lumen. Okay, now that turns out to be interesting. So what happens? How does the hydrogen peroxide signal get to the nucleus? Here's a closer, just a little bit of work I did with Emily Breeze at Warwick, who had a nice verified marker for ER, an RFP marker for ER. And here you can see the reconstruction of a series of Z stacks. And here you can see that there is a contiguousness of the perinuclear space with the ER lumen, and of course there's chloroplasts associated with that. Now, that would mean, okay, yeah, we'll go on for that. Oh, yeah. Just another point is that chloroplast associated with the nucleus is not some rarity of Nicotian iron or bentham iron abaxial cells. Every plant cell you look at has them in every species. And it's been well noted for a number. There's not a huge literature on it, surprisingly, but there's certainly been scored well very often. Okay, so you see very good connectivity. You see associations. One of the things we've been doing is that you can exploit a movement of chloroplasts away from incident light, which is phototropin driven. And here you see it, and this is looking at the bottom of, from the abaxial surface of the leaf, looking upwards into the leaf, into the base of the palisade. So what you see is some changes going on when the light is applied at an instant angle. Here before the highlight, here after the highlight, you can see there's some distribution going on. And here you can see attachments, chloroplast to nucleus. Now, the interesting point to make is that chloroplasts move away from instant light, but nuclei cannot do it on their own. They're dragged by the chloroplasts. The chloroplasts and the nuclei form a form of caravanserai and move everything down. So therefore, it's possible to look for mutants which are detached from each other, chloroplast to nucleus. And we started to do this in some work, a little bit of work, and this is the story we did. No transgenics here, but we've used mutants that are implicated in chloroplast development. So, for example, chloroplast division, chloroplast association with the plasma membrane, and so on. And we found two. This is one of them. The other one is well-known one called CHOP1, chloroplast unusually position 1. This one, Park 6, is a paralog of ARC 6, which is implicated in plastic division. And here we do a scoring where we apply an instant highlight. And if you look at col 0 here, and you look at in maximum intensity projections, you stain the nuclei with brains gone. You stain the nuclei. And of course, you've got the chloroplasts there as well. Then what you see is the chloroplasts move, and you can see that there are these bright nuclei also associated with that. But in this mutant, you can see that the chloroplasts still move, but they've left most of the chloroplast, the nuclei behind. So what we've done now is taken those two mutants, cross them to all those biosensor lines that I mentioned, right, to test the notion, how far does the hydrogen peroxide accumulation depend upon an active association between the chloroplasts and the nucleus. So that's as far as I could take you on that, except that, again, our favorite marker gene does show suppression of induction in response to highlight, Part 6 and also in show. Another point about the spatial component business is the different tissue you look at. So this is bundle sheath cells with bundle sheath specific expression compared with mesophyll. And the only point I want to make here is look at the difference in the starting oxidation state of those probes. This is the bundle sheath. This is the mesophyll. So chloroplasts in different tissues have very different oxidation states, implying different physiology and different roles. And in fact, you can see the changes here of H2O2 response, and you can see that the pattern is different. Does that reflect signaling? Yes, it does, because highlight ABA signaling is involved in highlight responses, but we can only show that in bundle sheath cells. In mesophyll cells, it doesn't appear to play a role. So putting all that together, sensory cells, sensory plastids, generally are smaller if you're looking out for them. 70% smaller than mesophyll chloroplasts on the whole in epidermis, vascular parenchyma, bundle sheath. There's lesser numbers of them generally in the cells, right? There's varying photosynthetic competence, and it also varies from species to species. But if you look at Arabidopsis, for example, trichomleucoplast, nothing. Bundle sheath cells and guard cells are photosynthetically competent. But there's PS2 light absorption activity in epidermal pavement cells and possibly in vascular parenchyma. Okay, this is all done by chlorophyll fluorescence imaging. Okay. But if you look in, no, that's it. So, but if you look at in abaxial tobacco cells, for example, the chloroplasts are photosynthetically active more so than in equivalent tissue in Arabidopsis. So there's a context there for all of this to operate, and in fact we can re-look at our data in the context of this kind of information. So what we're looking at, and this rather odd diagram was stuck into the grant proposal at the last minute. So we have, in fact, a pathway of hydrogen peroxide being made in the chloroplast stroma, which we postulate, in the case of those chloroplasts associated with the nucleus, crosses very rapidly into the nucleus through these membranes, probably facilitated by aquaporins, okay, into there. But what you have to bear in mind, there's also an evolutionarily conserved H2O2 signaling pathway that probably deals with most of the hydrogen peroxide signaling elsewhere in the cell. The ER, the mitochondria, even chloroplasts are not attached to the nucleus. But this is the thioperoxidase base system, which is present in us. It's impressive in all pretty much aerobic eukaryotes. Okay. And our argument is that, in fact, therefore, there are two pathways operating. One, the chloroplast, to communication to the nucleus, which is unique to plant cells and to algal cells, and it's evolved from algal or cyanobacterial systems. And then a second one, which is conserved across the eukarya, which involves the transmission of redox signals by oxidized thioperoxides, which could go anywhere but interact with target proteins by onward signaling in the nucleus, for example, but will go through a different route. The key of all of this is what's going on in that perinuclear space in the nucleus. And that poses some major challenges from a biosensor perspective. It's highly oxidizing environment. And if you put something like hyper in there, or those RhoGFP2 based probes, all they do is sit there fully oxidized. Nothing happens. So you need to build another kind of probe. Fortunately, some clever structural biochemists in Cambridge came up with a way of doing this, which we've modified to be able to hyper. What you can do is you can put in what's known as a diversionary redox active cysteine. So this hasn't got two redox active cysteines, it's got three. Two of them associated with the optical behavior of the hyper, but one of them is not. You know, that one can interact with the oxidizing systems within the ER lumen, which is in fact protein disulfide isomerase. It's very active. And what happens then is that you will lose some of your probe. Your probe will end up being fully oxidized. But the argument goes, you'll still have a population of probe left, which is still active to report hydrogen peroxide. And this works in mammalian cells. So there's been mammalian cells subjected to stresses that are associated with insulin and glucose starvation in HeLa cells and also pancreatic cells, showing that you can see changes in hydrogen peroxide concentrations within the ER lumen and in the perinuclear space as associated with this. And the triper, as it's now called, is reporting that. We've built a plant version and it's ready to go. And we're about to test it in Nicosia. When I get back, there's only me now, I'm retired, so I have to do it. So I'm doing this until the grant starts. So that's one, but you'll see that the optical characteristics are very different on that probe. So there's something to learn about how that probe actually works. Finally, just a last minute or two, is to try to build into why you might be to fuel it. Well, it doesn't matter whether what you postulated about the hydrogen peroxide, it's still probably got to be converted to some other type of signal in order to interact with regulatory proteins. What we do know from what you might call a lot of failed genetics is that the system is highly redundant. So as soon as the hydrogen peroxide probably enters the nucleus, it probably disperses into a large number of different targets very quickly, like rabbits running down holes. And we assume, for the moment, that it's still a thiol-based signaling system. Now, the classic one is something like this. Glutathione peroxidases get oxidized. They then can pass on oxidizing equivalent onto say a regulatory protein, which is then activated. This is very similar to what goes on in yeast that Michel Toledano described many years ago. And then the thyrodoxins will re-reduce them via NTR to the cellular and a DPH pool. Fine. And the same with peroxyredoxins as well as glutathione peroxidases, which are both classed as thiol peroxidases. But there is also another possibility, and this is one that's been raised more recently, is that the thyrodoxin can become oxidized. So the PRXs fix the hydrogen peroxide or other peroxide, they become oxidized, and they can oxidize a regulatory protein directly. Okay, now all that means at most is, oh my goodness, all the peroxidases can do it and the thyrodoxins can. What's the implications of that? Well, we don't know in plants yet, but if you look in yeast, and this is a really nice paper that appeared in 2011, and I take inspiration from it. In yeast, let alone in plants, there are 8 thiol peroxidase genes. And you have to knock out all 8 before you can remove the hydrogen peroxide transcriptome. And the cells are viable, amazingly. Furthermore, if you challenge oxidative stress with diamide, something that doesn't involve hydrogen peroxide, you get a full normal transcriptome response. So it's very specific. So you have to take out virtually all 8. Well, the implications of this are really quite important from a plant perspective. First of all, there has to be, if you put one of those GPXs back, you can restore quite substantial amounts of that H2O2 transcriptome. Okay, so, but if the TPX is in a mitochondrion, how's it working in dealing with signaling from other hydrogen peroxide with the sources? If it's in the nucleus, same, or it's in the cytosol, same. Implies there's a lot of movement of the proteins. Secondly, okay, that's in yeast, and look how difficult it is. Plants have about 20 TPXs, and thyrodoxins, 40 or so. Okay, you know, it's just a non-starter from a genetic perspective. I wish I knew this 20 years ago when I had more energy, because I wouldn't have done some of the things I did. So. The other thing that's interesting from yeast, and it's a good source to go to for inspiration, and this is where, if one takes a thyrodoxin out, then one gets various effects on the oxidation state of some proteins in the pool. But if one takes that equivalent to the NTR out, the thyrodoxin reductase here, then you get these oxidized thyrodoxins coming in, and they can behave the souped up signaling molecules then, doing lots of transmission. So the thought that comes to me, the type of thought that comes to doing pandemic lockdowns, is that what if in a wild type situation, say the wild type plant, a thyrodoxin reductase and NTR in the nucleus could move temporarily out and create a microdomain which was NTR deficient. Then those thyrodoxins would suddenly start behaving like this. So in other words, there's another spatial level to uncover. And this, I'll finish with this, because this is where we are, and this is why Jean-Philippe now, approach Jean-Philippe. NTR sits at this interface between this thiol regulation and the rest, really, of cellular metabolism. It's very key. And it's also the Achilles heel of the system from a genetic perspective. There's only 2 genes in Arabidopsis, NTRA, NTRB for mitochondria, the cytosol, and the nucleus. And there's one in the chloroplast we can sort of discount, NTRC. So there's only 3, not billion, like with everything else. So our way into the system is to manipulate this. And this is what this has been, our visit has been about here, is to try and see if we can get into the system, and maybe we get lucky that these TRXs are the next step. But even if they're not, then it's the thiol peroxidases after that. Okay, so that gives us a way into this network. We should probably use protein biochemistry rather than genetics. And with that, I thank you for listening, and I thank also my co-conspirators before Jean-Philippe, who's not on the slide, but I apologize for that. But we have Maria Expositor Rodriguez, who did an excellent postdoc with me, but now works for AstraZeneca. He's been lost to the biomedical world now. Philippe Luzier, who is our expert at all things confocal. And then we have some structural biochemists here, Mike Hoff and Brandon Reeder, who will help in the redesign of that ORP probe. Greg Brook, who's doing this equivalent work. We're running the same sort of ideas, but on the alien and cancer side, as well as the plant side. Me was Frets in the background. And then I acknowledge a previous, but now no longer existing, collaboration with Nick Smirnoff when we were funded by our first BBSRC grant in this area some years ago. And with that, I thank you for your listening.